wish you were already gone. I would be very offended, however, as these clipboards go back, if you sign it and immediately walk out, okay? I'll be watching, all right? So anyway, hopefully not very many of you will do that. What they asked me to talk about today is some of the new soil biology tests that are being used as part of the assessment of soil health. But I'm going to start with some of the older soil biology information that we have, and that's earthworms. Many of you who have been around for a long time know that I did a lot of work on earthworms back 20 to 30 years ago. We haven't talked about it as much recently, so it's kind of recently come to my attention that a lot of the younger folks don't know that Purdue ever did anything on earthworms or that we know anything on earthworms. So I'm going to try to talk a little bit about that. And those of you who have heard me talk about earthworms probably are ready for a refresher. And then we'll talk about some of the new soil biology tests. So soil biology and soil health tests. I like to use this picture from a National Geographic article on the soil that was out, I don't know, 20 years ago or so, just as a reminder that even though today I'm going to talk mostly about earthworms, that there are many other creatures in the soil that are affecting the soil. Yeah, great, thank you, Paul. We have creatures that are much smaller than earthworms, microorganisms, protozoa, nematodes, good and bad nematodes, and we have creatures, of course, that are much larger than earthworms. But earthworms, because we can more easily see them and count them, we tend to a lot of times focus on them. Why are we interested in soil biology or soil ecology? Because it has a lot of important impacts. Nutrient cycling is mediated to a large extent by organisms that are working in the soil. Organic matter mixing is very much a function of organisms working in the soil, especially when you are not doing a full inversion tillage operation. So the decomposition of organic matter and the stabilization of organic matter is all related to organisms. Soil physical properties are very much affected by organisms. Pest-predator interactions, of course, Christian was just talking about some of those. So all of those are just some of the examples of why we're interested in soil ecology. So there are a variety of ways that soil organisms get classified. And I'm going to talk about some of these new soil biology tests, but in order to put them into context, we need to think about how people categorize the different groups of organisms. So sometimes we simply characterize them by size. How big or small are they? Okay, and you're probably most familiar with the very small ones, the bacteria, and the much larger ones, like earthworms. But there's a whole group in between, and I'll just talk briefly about them. Sometimes we characterize them by where do they live. Do they live in the water-filled pore space in the soil, or do they live in the air-filled pore space? Sometimes we characterize them by their function or their trophic level. Where are they on the food chain? Okay. Sometimes we characterize them genetically with some of the modern techniques with DNA and so on, which are pretty much in their infancy and their use in soil biology. The way I'm going to talk about them a little bit is looking at both their size and their trophic level, or where are they on the food chain. And just very briefly, so don't worry about that. First of all, the ones that you hear the most about are the microorganisms, or the microflora, the bacteria and the fungi, okay? Then we have the next level up, and that would be the microfauna, okay, fauna, animals, of course, and that's the nematodes and the protozoa. And of course, nematodes, there's a lot of good nematodes and some bad nematodes. You're probably more familiar with the bad ones than you are with the good ones, but those organisms feed on and predate a lot of the bacteria. Then we have the mesofauna, or the in-between size ones, the springtails and the mites and the enchiotrades. And then we have the macrofauna, which are very visible to the naked eye, and that would be earthworms, termites, ants, and so on. So again, I suspect that most of you are familiar with each end, the very small and the very large, but not so much with those in between. Another way of looking at organisms is by their function. And one terminology that I found pretty helpful was from Patrick Lebel, a soil biologist in France. when he was writing about the functional guilds of the soil fauna. And again, the one that you're most familiar with is that first one, the micro food webs. And that's basically where the microorganisms are linked together with the folks that are predating on them, okay? So the organisms that are feeding on the bacteria and fungi. So it's kind of a food web, and that's very much related to nutrient cycling. The next level up would be classified as the litter transformers. And so these are the ones that a lot of times in our agricultural systems we don't pay a whole lot of attention to, but as we go to more reduced tillage systems, they can become more important. And so these would be some of the larger beetles and so on, dung beetles as an example. They physically fragment litter residues and they ingest them. And then their fecal pellets contain mostly the organic material, very little mineral soil. They don't really eat very much mineral soil. And then after the fungi and bacteria have decomposed some of the material in those pellets, then the beetles may re-ingest them and get some nutrition from them. They don't do a whole lot for soil structure, but they do help break down plant residues or litter. And then the last group, the ecosystem engineers, which are my favorite, and they do a lot of things like we kind of think of engineers doing, right? They build tunnels and bridges, and they make holes in the ground, and they move things around. And so the earthworms would be the ones that we're most familiar with, but if you're in tropical soils, termites would be a very important part of that. So they're digging burrows, they're ingesting litter and mineral soil, and their fecal pellets or casts are a mix of mineral soil and organic materials, and they can really help improve soil structure. Okay, so the earthworms, an obvious ecosystem engineer, building tunnels and mixing organic materials into the soil. I'm going to talk just a little bit about earthworm ecology. There are thousands of species of earthworms in the world, but we don't need to know all of those. We just need to think about, first of all, what are the major behavioral groupings? And in our temperate climate here, we have two major groupings and a third one I'll briefly mention. But the deep burrowing earthworms, okay, which we know as night crawlers, they dig a deep burrow, and that might go 3 feet deep, might be 4 feet, 5 feet, maybe even 6 feet deep, depending on the soil. The night crawler lives in that burrow more or less permanently during its lifetime, and it pulls residues down into the mouth of its burrow, and I'll show you some pictures of that in a minute. It feeds on those residues and then it pulls more residues down. So it pretty much stays more or less in place for most of its life. Then we have the second group. Oh, and there's only one species in the glaciated part of North America. There's only one species of deep burrowing earthworm, and that's what we call the night crawler, or Lumbricus terrestris for the scientific name. We then have shallow dwelling worms. They live primarily in the topsoil, top 8, 10, 12 inches. And there are multiple species of them, and they are called by different common names depending on what state or what county you're from. Red worms, gray worms, fish worms. We have several different species of them. So a typical Indiana field might have 2 or 3 different species of red worm. If you go to a wooded area, you might have 5 or 6 different species of them. But they pretty much are burrowing around in the topsoil. They don't have a permanent burrow, so they're basically feeding and excreting casts out the back end as they go, and they're feeding on both mineral soil and organic material as they go. Okay, so the red worms, if any of you have been digging soil pits in the middle of the summer, you might run across these worms that are all curled up. Basically, the red worms are active during the spring. And then in the summer, when the soil starts to get dry and hot, they move down a little bit deeper. And it might be 15 inches, maybe even 18 inches. And they curl up in a little ball like this, and sometimes they're even in a knot. And they secrete a mucus to try to keep from drying out, and that's how they spend the summer, in that resting stage. So if you're digging a soil pit because you're repairing tile or you're preparing for a field day, and you have earthworms, quite often you will disrupt a few of them. We did that this week on our soil health training. We were digging a trench, and we got quite a few worms that were disturbed from their summer nap by our activity. Then in the fall, when the soil starts to wet up again and cool off again, then they become active, and then in the winter they go into a hibernation stage. Okay, the night crawlers are a little different because they have a permanent burrow and it's deep. They don't necessarily go into a complete resting stage in the summer. Many of you who have worked in no-till fields or have no-till in some of your operation have probably seen this, but these are middens. This happens to be in a corn-soybean rotation in the spring after soybeans. And you can see all these little piles, and these are not piles that the water just washed there. Okay, these are all done by the night crawler. A little bit closer view here, you can see we've got corn stalks from 2 years ago and soybean residue and some of the cast material from the night crawler and some soil all kind of mounded up. And some of that residue has been pulled down into the mouth of the burrow. Now I've shown you a picture here where it's very, very obvious where the middens are, but in many of the no-till fields I've been to, it's not quite that easy to see them because there's still a lot of residue. So you have to kind of carefully lift the residue up and be looking for the middens underneath. So we've got a midden there, and if you carefully kind of pull the plug on that, you can see the residues that have been drawn down into the mouth of the burrow, okay? Typically that's only a couple of inches, and they pull it down in there, and because it's a moist environment, the bacteria and the fungi start working on it, and then the night crawlers feed on the fungi and bacteria as well as the decomposing residue. I'm really fascinated with middens, so I can, no matter how many of them I've seen, I can go out and spend hours just kind of looking at middens and trying to pull the plug and take pictures of them. And so this one was a particularly long piece of corn residue when I kind of inverted the midden that had been pulled down into the burrow, so it was pretty impressive. Okay, so what are some of the earthworm benefits? They do their engineering thing. They create large channels, which can be really important for getting water and air down into the soil and for roots to develop. They improve soil tilt or soil structure, and they incorporate organic matter or nutrients into the soil. This was a soil pit in Benton County a number of years ago, a no-till field. And I did my little archaeological excavation here, and you can see a night crawler channel going well down into the subsoil. And you can imagine what kind of an impact that has on getting water flow into the soil profile on heavy rainstorm conditions. Another feature of the night crawler channel is that at least part of that channel is lined with the cast material from the night crawler. Okay, so the cast material again is the excrement, and so it's a mix of mineral soil as well as partially decomposed organic materials. Okay, so that material is higher in nutrient availability than the soil from which it came because it's releasing some of the nutrients from the organic material or the residues. So you can imagine that if a root happens to find one of those channels, it tends to proliferate in it. This was a subsoil in a no-till field in central Illinois, and you can see the corn roots were really proliferating. You can maybe see some of the dark cast material there in that channel. Basically, those roots are proliferating in there for two reasons. One is there's a lot of extra nutrients, and two is it's an easy place to grow. So we can have some impact on our rooting by having a higher population of night crawlers. In my opinion, earthworms and other soil fauna, beetles and so on, are much more important in a reduced tillage system, and in particular a no-till system, than they ever were in a moldboard plow system. So when you think back when everybody was moldboard plowing, we kind of forgot about soil biology pretty much altogether because you had a moldboard plow to mix everything up. And now if we have less tillage, we need the organisms more in order to do some of that work that a tillage implement did. and they can do other parts better than the tillage implement because they are secreting mucus and aggregating the soil. Okay, greater populations of earthworms and other fauna, we would say, are going to increase the diversity of the underground food web and the complexity of the underground food web. Okay, is this good or bad? So that's part of the question. Is greater biological complexity and diversity good? We usually say, with what we know about most systems, that the more complex that system is, the more different organisms that you have in there, that's a benefit for the stability and resiliency of the system, okay? But there's very little research that's actually studied it because in part it's very, very complex. So in the soil biology realm, there's not a whole lot of data. We generally assume that it's better, and why? Well, one of the reasons is the ecological concept of redundancy. If you have more than one organism that can do a job, if you have a stress that kills off the one organism, but there's still somebody else there that can do that job, then you're more resilient, right? You can still go on and have that function performed. And that's basically what folks think is happening with the more complex soil biology, is now we have 3 or 4 or 5 organisms, or 3 or 4 or 500 organisms that can do a particular job. So if we wipe out 100 different kinds of species, but we still have another 100 that can do the same job, then we're much more resilient to stresses. So that's the thought. But it's still, I would say, relatively not well known how important that is. So we do need research on these kinds of impacts, as well as how do we increase biological diversity, and that's part of what I'm going to get to here with our Conservation Innovation Grant, CIG. And is there some optimum level of diversity? I mean, we say more is better probably up to a point. Okay, and at some point we probably have enough redundancy and we don't need it. But I would suggest that in most of our agricultural systems, we are nowhere close to having enough of that diversity. Okay, how does tillage impact organisms? The physical disruption and the residue burial are the chief things that have an impact, but these other 3 things, the surface area contact between the organisms and the food source, and their effect on water and temperature, and their effect on pore space and continuity are also important. All right, this was an article that kind of reviewed over 100 different studies that had been done on tillage versus no tillage on some aspect of soil biology. It might have been earthworms, it might have been mites, it might have been springtails, it might have been bacteria and fungi, it might have been a group of those. Most of the organism groups had higher abundance in no-till. Tillage had a greater negative effect on larger organisms than smaller. Okay, so the tillage effect had a worse effect on night crawlers than it had on bacteria, for example. Okay, and again, you can imagine if you're disturbing the soil and mixing the residue and cutting organisms up, it's going to have more of an effect than on the tiny, tiny microscopic organisms. There were variations in response, and certainly there are some organisms that ecologically are ready to go in a disturbed system, right? Any kind of ecology, when we have a fire, we have certain plants that are ready to come into that disturbed system. So the same thing with soil organisms. There are some organisms that do better right after tillage, but most of the diversity of organisms that we're looking at do better with less disturbance. So when we do earthworm work, we go out and count the earthworms in the spring when they're most active. So this was from some of our work in April, where we basically dig up a known volume of soil and we sort through the soil by hand and pick out the worms and count them. And if any of you want to volunteer to help me the next time, that's fine, because every time that I do this, my students or whoever's working for me at the time, if they've done it one year, they try to make themselves scarce the next year, okay? So always looking for volunteers to help with that. It's very tedious work, but, and then the middens, again, if there's a lot of residue here, I'm on my hands and knees counting middens because you kind of have to lift the residue up, and so we we block off a known area, and then I count the middens in that known area. Okay, we've got quite a lot of data. Again, we did a lot more work on this back in the 80s and early 90s. I haven't done as much recently, but we're doing some more again now with our new project. But this is just giving you an example. This was in southeastern Indiana, Jennings County, in April in a corn-soybean rotation. And this is earthworms per square meter or per square yard. And the chisel system in that case was on the order of 50 earthworms per square meter. And these were all redworms. This particular field at that time did not have any night crawlers in it. Our ridge till and our no-till systems were in the order of 130 to 210 earthworms per square meter. So somewhere 3 to 4 times as many earthworms where we had much less tillage than where we had tillage. In this particular project, we didn't actually count the moldboard plow, but from other work, I would know that that would be even less because there was less residue cover over the winter. Okay, we did several studies on Purdue farms, and then we went on our safari across the state of Indiana and Illinois. What I wanted to do was get out on farmers' fields and see if we were getting similar kinds of results. So the main results here, we had 14 pairs of till and no-till fields, and NRCS helped me with finding these pairs on the same soil type, either across the fence or across the road from each other. And basically, on those 14 pairs of sites, the shallow dwelling worms, we had higher populations in the no-till compared to the conventional on 8 of the 14 pairs of sites. And the higher populations ranged anywhere from 25% higher to 10 times higher, depending on the site. 4 of the sites had approximately equal populations in no-till and conventional, and 2 of the sites had less in no-till than conventional, but those sites actually had very low populations in both. So, and they had also not been in no-till as long. They had only been in no-till for 3 years. So basically, those results pretty much substantiated what we had seen in our research and what other people had seen in research in Europe, Australia, and New Zealand, in that no-till generally is going to have higher populations than conventional. The night crawlers were even more dramatic because their ecological niche is to have residues at the soil surface and pull them down into the mouth of their burrow. So if you're doing tillage, you're going to destroy that ecological niche, basically, and destroy the top part of their burrow. So 9 of our 14 no-till sites had evidence of middens and night crawlers, and only 3 of the conventional sites did. So again, the night crawlers are much more sensitive to the tillage than are the redworms, but both of them do better where you have no-till than where you have conventional till. Okay, before I get into the new soil biology tests, I just want to tell you a few other kind of summary things related to worms. If you want to count them, the best time to do it is in the spring when they're most active. If you're doing the shallow dwellers and you want actual numbers as opposed to just a quick look-see, you would want to shovel a known area about 10 inches deep, because if you're doing it in the spring, that's primarily where they're working. You would sort through the soil by hand and count the worms. If you're looking for night crawlers, if you start digging the soil, of course, you all know if you've ever tried to catch them for fishing, that as soon as you start digging, they're down the bottom of their burrow. So usually what I've come to do now is just count middens, okay, as an indicator. And it's a pretty good indicator of their population. And you need to repeat those observations because earthworm populations are spotty, okay? They're not uniformly distributed across the whole field. So I would say a minimum of 5 spots in the field if you want to get an actual estimate of populations. So we're interested in soil health and soil quality, and sometimes people want to use earthworms as an indicator organism. And I would say this could go both ways. If you have a lot of earthworms, whether it's redworms or night crawlers or both, if you have a lot of earthworms in your field, that probably indicates that you have high quality soil, good health. If you have few earthworms or none, it doesn't necessarily mean that it's bad quality. There are other organisms that are important too, and sometimes the reason there's no worms there is because they never had a chance to colonize. So there are some fields where we don't have them, not because the soil's bad, but because they never actually had a way of getting to that field. Different species do different things, and so for indicators, you kind of want to know, well, which organism do I want to count? So that makes it a little bit difficult. It is hard to get accurate counts. They are seasonal. You can't just decide in the middle of July you're going to go do an earthworm count. I would not recommend that. Okay, that's not when their populations are at their maximum. They can vary with short-term weather variations. They're patchy, and you saw that they're somewhat labor-intensive to get good counts. We don't necessarily know how many we need, and we don't necessarily know if they had an opportunity to colonize. So this just means that earthworms as indicators have their limitations, okay? When people press me and say, well, how many do I need in order to have a good quality soil? These are numbers that I know are realistic and achievable based on my experience with corn soybean type rotations in Indiana. Certainly higher numbers are possible and are even better. But if we're talking about a corn soybean system, I know that our shallow dwelling worms, we can find them in the 100 to 200 per square meter range, which is 10 to 20 per square foot. Or if you're one of these people that likes to throw out all these big numbers about how many worms you have per acre, okay, it's 400,000 to 800,000 per acre, okay? The night crawlers, you're going to have fewer of them. They're bigger animals, slower to reproduce. You don't need as many. They feed from a larger area. So those numbers, if you have 10 to 20 per square meter or 1 to 2 per square foot, that's a really good number. They can get higher than that, but I would be quite happy with a field that had that kind of level of night crawler. But the year-to-year variation is going to be quite large. All right, so I'm going to switch gears now. That was kind of the earthworm stuff, which we have done for quite a while. I think maybe on the materials that you got for this, there should be an earthworm extension publication that I wrote a number of years ago, which would be kind of a good reference on earthworm ecology. It's also on the web. So let's take a look at some of the new soil biology tests that people are proposing that are commercially available and that people have been asking folks like Barry Fisher and Dan Towery and Hans Koch and myself, you know, oh, there's these new soil biology tests that claim to represent soil health. How good are they? What do they mean? Do they tell me anything that can be useful to my management? And none of us have any experience with these because they are new, they are pricey, and the interpretations at this point are kind of in their infancy. So we decided as part of this CIG or Conservation Innovation Grant that we're doing around the state to get some experience with them. So we're taking samples from 12 cooperating farmers as well as from some Purdue sites, and we are doing these 4 tests, and we hope that over the course of the 3 years we'll learn something about how useful these tests are. The first 3 are really what I would call biological tests, and then the Cornell test has a combination of biology, physics, and chemistry, and we'll go through those, okay? I want to point out here, this bullet here, interpretation of these are in their infancy, and there's no standard like with the soil fertility test. So you all know what a high soil phosphorus or soil potassium test is. There's years and years of research that kind of document what those levels should be. You might debate them on some points, but basically those are fairly well established. For soil biology tests, there is no such thing yet. Okay, and that's going to be a while. That's part of the reason that we're doing this study. Okay, the first one of these is the PLFA, phospholipid fatty acid, which is done by Ward Laboratories in Nebraska. Jill Clapperton, who's a soil biologist originally from Canada, now in Montana, was the person who helped develop some of the interpretations for this test. The phospholipid fatty acids are in living cell membranes. They decompose very rapidly after the cells die, okay? So they are a snapshot of the currently living cells. You're going to see a theme here that almost all of these are a snapshot. So right now, today, when I take this sample, this is how many cells are living, okay? It doesn't tell me anything about what was living a week ago or a week from now, okay? There is some ability to characterize broad groups of organisms. protozoa, fungi, nematodes, gram-negative and gram-positive bacteria. There are some very general interpretations that are given. Fungal to bacterial ratios, you've probably heard before that no-till systems tend to have a greater amount of fungi, not greater fungi than bacteria, but have a higher ratio of fungus to bacteria because of the non-disturbance, and that helps with soil aggregation. So these tests can give us that. They can be useful to compare sites. So today I want to compare this till versus no-till, or this manure versus this non-manure, or this cover crop versus this non-cover crop. Or take this site and compare it this year to next year at the same time, to the following year at the same time. But they're going to be very difficult to interpret across wide areas, we think. And the sampling protocol is extremely important, because if this is for living cells, and I take the sample and I throw it in the truck, and I let it sit there over the weekend and it's hot, there are not going to be very many living cells left, right? So we had to be very particular about putting the samples on ice, shipping them overnight, and so on. Okay, for those of you who are interested, this is an example of an output that they gave us. I just want to point out a couple things, because again, I don't fully know how to interpret these. We had a soil biologist in our department work with us a couple weeks ago to do an initial explanation of some of this, but I hope in about a year we'll be able to tell you a lot more, maybe sometime in the winter even. The first number is this total living microbial biomass, 1,100 nanograms per gram, so 1,100 parts per billion. So that's basically giving us our total living biomass. So that's one number that's probably a pretty good number. Doesn't tell us much about diversity. The second number, which I did not circle, is a diversity index. I don't know exactly what that means yet, other than it's supposed to relate to some degree of diversity. And then over here on the left, it breaks that 1,100 parts per billion down into different groups. Bacteria, gram-negative, gram-positive, rhizobia, actinomycetes, different kinds of fungi, arbuscular mycorrhizal, saprophytes, which are decomposers, protozoa. And for those of you who, in the front who can see the number, undifferentiated, that means we don't know what they are. 61% of my sample. Okay? So I find that a little disconcerting. All right? So it's telling us something about 40%, but the other 60% they don't know anything about it. And this is a common thing in soil microbiology, that there's a lot of organisms that we don't know what they are, we can't culture them, we can't grow them when they're not in the soil, and so it's very difficult to know exactly what they're doing. Okay? So that's one example. And then on the second page, I know you can't read this at all, I'm simply circling here that they're telling us the ratio of fungi to bacteria, they're telling us ratio of predator to prey, and all these different ratios, they're giving us some very basic qualitative interpretation. Okay, the second one, which also has a soil biologist who originated these, Dr. Eileen Ingham, who was at Oregon State and now pretty much does private work with this company. They use microscopic techniques, in other words, looking under a microscope to determine bacteria, both active and total, fungi, active and total, protozoa, and if you wanted to spend additional money, it would be nematodes. Oh, just for your information, this PLFA one is about $37 a sample. This Earth-4 one is about $110 a sample. So you can see that we're talking pretty pricey samples, and that's why we're somewhat limited. So it measures bacteria, fungi, protozoa. It measures the biomass of those groups. It does not give you specific organisms. Again, it's a snapshot. Who's there today? The interpretations are broad, and the sampling protocol very, very important. Again, this is just an example. We have real numbers. It's pointing out here active and total bacteria, active and total fungi. It's telling me whether they think this is below the range or above the range or in the range that they would consider to be good. Okay? Down here it's talking about some ratios of fungi to bacteria and things like that. And again, they give us some very general interpretations. what I noticed when I quickly looked at some of the results that we got this summer is that a lot of them say foods may be required. Okay? So in other words, we're saying that more food, more organic materials are needed in order to bump up the soil biology. Okay, the third test is the Solvita CO2 burst. Essentially here, what they're doing is they're saying, okay, we're going to take the soil, we're going to dry it, and then we're going to re-wet it. And whenever we dry a soil and then re-wet it, we get a burst of carbon dioxide because some of the organisms died and some of the organisms just kind of went to sleep. And when you re-wet the soil, there's a flush of food and a flush of activity. So this is basically saying that the bigger the flush of activity, the more microbes that we had in the soil. Okay? So again, it relates to the microbial biomass present at the time, but because we're drying everything and kind of putting them in suspended animation, the sampling protocol is not nearly as important. This is also Dr. Rick Haney in Arizona, in Texas, and he also has another test where he extracts nutrients, but he uses different extractants than our typical soil tests. So basically he's extracting with water and with a weak acid, and these are supposed to be more similar to what a root would feel today. Not necessarily what's going to be available over the whole season, but what it would feel today. I know you can't read any of this. This is not even all of the output, but essentially part of what he's giving us is a carbon-nitrogen ratio of what gets extracted with the water. What kind of cover crop mix would they recommend based on what that carbon-nitrogen ratio is? I don't necessarily buy any of this yet. That remains to be seen, but it's an attempt, it's their interpretation of what they think might be needed to improve the soil. The last one I'm more comfortable with simply because I know more about it, and that's the Cornell Soil Health Test, because it includes not only biology but also physics and chemistry. And I work on soil physical properties, so it includes tests for all of those. It's based on extensive work actually over the last 20 years, where they've ARS and NRCS have tried to come up with these curves that relate aggregate stability to yield, or bulk density to yield, and so on. The Cornell folks actually originally evaluated 39 different tests to see which ones would be most sensitive, but also reproducible, and that would be best indicators, because they wanted something that was reasonable to measure, but that would indicate crop productivity. Each measure is rated on a 0 to 100 scale, and then those are added together for a composite score. So the sampling protocol on this is also important. You can't read the specific things here, but you can see we've got red, yellow, and green colors. So what we have here is the indicators. These indicators up top are the physical indicators, then we have biological indicators, and we have chemical indicators. The first column, the white column here, is the actual measurement of that property. So for example, their aggregate stability on this sample was 18%. Their available water capacity was 18%. Their surface hardness and PSI was 348 PSI. So those are the actual measurements. And then what they do is they assign a rating, okay? So an aggregate stability of 18% has a rating between 0 and 100, in this case happens to be 18. The available water capacity of 18% has a rating of 64 on a 100-point scale, okay? You can see here there's a lot of red. So, and it tells you, okay, 18% aggregate stability and a rating of 18. That's bad because you're going to have aeration problems, infiltration problems, and rooting problems, okay? The surface hardness is bad because you're going to have rooting and water transmission problems, okay? The organic matter here has a rating of 9 out of 100, and it's basically saying you're going to have a problem with energy storage or carbon for the microbes. So it's giving a rating, which you can, agree with or disagree with. At least the column that's in the white here is the true reading, aggregate stability 18%. They're telling me that's really bad. I would agree. It's really bad. So they're telling you why they think it's bad or why they think it's good. A red, green, and you'll notice that a lot of our soil, this is an example from their website, but the chemical properties are all green. They're all good. You know why? That's what we usually pay attention to, right? So we make sure that we've got adequate phosphorus and potassium and that our pH is right. So that's all in the good range, but our physical and biological are pretty bad. So then they sum up all these numbers and they have a total score of 43 out of 100. So that's considered low quality soil. So they basically are giving you that. All right, so what do you do with that? Well, this is basically giving you some suggestions about what you might do if you want to improve those things. And then if you start doing some things to improve your soil, this is something that you could come back the next year or a couple of years later at the same time of year, take the test again, and it would essentially be a way for you to monitor progress, hopefully progress, in improving your soil. Okay, so the soil biological tests are still experimental. There's a lot of organisms that we don't know how to measure, let alone what they do. Populations are changing all the time with moisture and temperature. Even in a healthy soil, you're going to have populations shifting as moisture and temperature shift. We don't exactly know how to interpret those values yet for practical applications, which is exactly the reason that we're trying to do all of these with a number of soils and fields around the state so we can learn how useful or not these may be, so we can answer some of the questions we keep getting. At this point, I'd say they're a relative measure to compare the similar sites at the same time or to compare one site over multiple times. And we will be obtaining some experience in our Conservation Innovation Grant project, which is going on around the state. So I think that's all I have. Thank you very much. I'll be happy to answer questions if anybody has any, and otherwise have a good weekend. Yes, sir.
A number of those tests had fungi, fungi, bacterial ratio. There's a lot of fungicides today.
So the question is the fungal to bacterial ratio. Currently, there's a lot of fungicides being used. Is that good, bad, or indifferent? Well, fungicides on the soil would be bad for the fungi. Many of the fungicides that we're spraying, of course, a lot of that's going on the plant. And if most of it is on the plant, by the time the plant actually dies and gets to the soil, a lot of that would be gone. But certainly, the less use of fungicides that we can have, the better, as far as fungi in the soil. Yes, sir.
As the soil is healthy